So let's talk about maintaining homeostasis using negative feedback loops. There's several syllabus objectives here. The first one is about using negative feedback loops to maintain homeostasis. We're going to look at stimulus response models and we're going to look at negative feedback loops. In future videos, we'll look at how the nervous and endocrine systems are involved in that. Second one's an easy one. It's talking about the different types of sensory receptors. And the third one is about analysing feedback control diagrams, and we'll look at a couple of examples to be able to do that. So first of all, homeostasis is about maintaining a constant internal environment. It's about balancing the conditions within an organism so that they are maintained around a certain point, which we call the set point. So looking at this tightrope walker here, if you imagine that it kind of loses his balance and his balance moves over towards this left-hand side, he needs to respond by shifting his weight to the right to bring his body weight back to the center or the set point. So his response needs to be in the opposite direction to the stimulus. It needs to reverse the stimulus. And that's what we call a negative feedback loop. So homeostasis is about maintaining a constant internal environment within an organism, and it does that using negative feedback loops. A negative feedback loop is where the response reverses the trend. So for example here, if we have a bowl and we have a ball in that bowl, if we were to roll the ball away from the midline or away from the set point, the response, and if we were to let go of the ball, the response is that the ball is going to roll back down to the midpoint. The response reverses the trend. Given that it's a ball, it might roll past, but then it's going to respond opposite to moving up this wall. So it just might oscillate a few times, and then it's going to stop here at this set point. That is a negative feedback loop. And that's what we use for homeostasis. Now, let's have a look at the alternative is if we were to take that bowl and turn it upside down and put a ball on top of that. If we were to push that ball, well, it's going to roll away from the midpoint and it's actually going to accelerate as it moves away. So the response increases or amplifies the trend. The trend was to push it this way, okay, and actually the response was to increase that. So the gravity made it increase. This is a positive feedback loop, and we don't use these in homeostasis. There are examples in the human body and other organisms that use positive feedback loops, but not in homeostasis, because homeostasis is about maintaining a constant internal environment. It's about bringing the conditions back to this set point at all times. Now, why is that important? Well, if you think about enzymes, we've already seen that enzymes have an optimum temperature, optimum pH, and other conditions in which they operate. Either side of that, you can see the rate of enzyme reaction decreases. So we want to maintain our temperature at the optimum temperature for our enzymes to operate. Moving away from that, it's not optimum. So homeostasis needs to be maintained for optimum performance of enzymes and other metabolic functions within an organism. And it does it using what we call a stimulus response model. So we have a stimulus, that's some sort of environmental energy, some sort of information. It could be an internal environment or an external environment, but it's some sort of information, it's energy that gets detected by a receptor. So in this case, we have the light or photoreceptors in the eye that detect that information, then gets sent through to the central nervous system for it to be processed, the control center. That control center interprets the information from the sensor, interprets that stimulus, and decides on a response. Then it sends that response through to what we call an effector that's actually going to make an effect or make a response. So it's the stimulus response process. So what are our different types of sensory receptors? This is what the syllabus objective says, is we need to know about these different types of sensory receptors and what they detect. So we've got chemoreceptors that detect things like pH, carbon dioxide in the blood, glucose, so the amount of glucose that's in the blood, as well as that, taste buds are chemoreceptors. They detect sweet, they detect salty, etc. Thermoreceptors detect temperature. So we're talking about temperature again. If we're talking about mammals, we're talking about temperature of the blood. Mechanoreceptors. These are in the skin, but they're also in the stomach and other parts of the body as well. And they detect things like stretch and pressure, touch, sound. So the hair receptors in the ear are actually mechanoreceptors, and they detect sound by vibration of the air. Position of the body. So we have receptors in our joints that determine the position of our joints, and we have other receptors in our ear called the vestibular system that detect the position of our head in space. Photoreceptors detect light in the eye. Nociceptors detect pain. So these are I'll just show you here, nociceptors. Okay, all of the others are something receptor. For noci, they're nociceptors. Okay, so they're all pretty straightforward. These are all the types of receptors. Now, the control center, it is often the brain, but not always. Sometimes it is the brain stem, or sometimes it's the spinal cord, and sometimes there's other organs, other glands like the pancreas. Now, in terms of effectors, I think this is really interesting, but there's only two types of effectors that we have in the body. They're either muscles or glands. Muscles respond by contracting. They have a neural stimulus, so from the nervous system, and their response is to contract. So that could be the heart muscle, it could be the diaphragm muscle for breathing, it could be in blood vessels like capillaries that contract and constrict the capillaries, or it could obviously be our skeletal muscles that move our body. And the other type of effector is glands. So the next thing we need to think about then is looking at these. So let's have a look at that. So the last syllabus objective is about analysing feedback control diagrams to identify the stimulus, receptor, control center, effectors, and communication pathways. Okay, so we're going to have a look at one that you probably know, and that is thermoregulation. So we have a receptor in our... So the control center for thermoregulation is the hypothalamus in the brain. And it sets a set point for our body temperature of 37 degrees. Now, if it detects that our body temperature increases, for example, it's a hot day, we might be exercising, it might be a really hot room, and our body temperature increases above 37 degrees. Then we have responses. The first one is sweating. Okay, so we excrete water through our excretory sweat glands onto the skin, and that water evaporates, taking heat with it. We have skin capillary vasodilation. Vaso means vessel, dilation means open. So it's the capillaries in the skin. They open up to bring more to bring more blood to the surface of the body, and with that blood brings heat. And the heat, when that when the sweat evaporates off, it takes that heat, and that heat can radiate off the body as well. So this is controlled by the nervous system that contracts or relaxes the muscles of the capillaries in the skin. Okay, the nervous system controls the sweat glands, and our hormones reduce our metabolism, reduce the amount of food that's being burnt to produce heat. The response here, so these responses decrease the stimulus, or in other words, they cool the body down such that the body temperature decreases back to the set point. So it's a negative feedback loop because the response decreases the stimulus. Now, we can think about what happens when the body is cold, when the body temperature decreases below the set point of 37 degrees. The hypothalamus in the brain detects that and does a number of responses. Firstly, well, it doesn't work very well in humans. The hairs on our skin create or become erect and causes goosebumps. Doesn't work very well in humans. Shivering, muscle contraction of the voluntary muscles of the skeletal muscles involuntary causes shivering. That produces more heat. Vasoconstriction of the skin capillaries closes those capillaries in the skin, keeping the heat in the blood closer to the center of the body, so reducing heat loss and increasing metabolism, so burning more food to produce heat. And these responses increase temperature, or they reverse that trend of decreased body temperature, so they increase the body temperature again. So that's an example of a negative feedback loop or diagram to demonstrate thermoregulation. So the syllabus objective is essentially to explain how the nervous and endocrine systems use negative feedback loops to coordinate responses to internal and external stimuli and to maintain homeostasis. So the stimulus can be internal or external. The receptors can be chemo, mechano, photoreceptors. The control center could be the nervous system or glands. The effectors can either only be muscles or glands. And then we can have two different types of response. A motor response, like shivering, or contracting the blood vessels, or a secretory response, like excreting sweat. Okay, and so that is the introduction to homeostasis and how we maintain it.